Let's look at model 4, molecular model 4. Okay, and in this model, we are going to look at the structure of chromosomes, the different types of chromosomes when it comes to packing, and let's talk about chromosome structure, function, and abnormalities. So first, look at the chromosome structure. Okay, and I'm sure we all know what a chromosome is. Chromosome is just like complex of DNA and proteins. Okay, that are compacted into to be able to fit into the nucleus. And when we say genome too, I'm sure we know what a genome is. Genome is like the genetic material, the total genetic material of an organism. Okay. So there is something called the C-value paradox. Okay, and this states that DNA content of an organism does not directly coincide with the complexity of that particular organism. So you having plenty DNA does not mean your genome is going to be complex, okay, than another organism with less DNA. An example is comparing amoeba and humans. We know humans have way more than amoeba. Okay, but look at the genome, haploid genome in base pairs in amoeba and humans. Amoeba has like over 290 billion, right? Humans have only 3 billion. Okay, so DNA content does not always it's not like directly proportional to your genome complexity. Okay, so let's look at how the DNA is packaged into the nucleus. So the DNA actually forms the chromosome, okay, complex around proteins. But how is this packaging done? Because the DNA is very long. Even not even talking about human chromosomes, prokaryote chromosomes are actually longer, and on their own they can't be fit in the bacterial cell. So whether prokaryote or eukaryote, the chromosome has to be folded or packaged in a way that it can fit into the cell. And in the case of eukaryotes, it's not even in the cell, but in the nucleus, which is smaller than the cell, right? So the packing should be efficient, and also should allow the packaging, okay, and also should allow accurate replication during the S phase of the cell cycle. So how is this done? And another challenge is, you see, you can have a really compacted or packaged DNA, but at any point in time, one particular piece of it has to be, depackaged for replication or expression to take place. So this picture here is showing a condensed chromosome, and it has a particular part of it that's called the chromosomal path. Okay, so that part of it is unpackaged at the moment, and every other every other part of the chromosome is condensed or compacted, right? But this particular place called the chromosomal path is being expressed at the moment, so it has to be unpacked. Okay, so let's look at viral genomes. So I'm going to talk about genomes of viruses, prokaryotes, and eukaryotes. Viruses are very brief, same for prokaryotes as well. So viruses, their genetic material is not actually certain. Okay, their genomes, and we all know viruses are particles and they require host cells for replication. Okay, what you need to know is viruses, there are two types of viruses: those with an envelope covering their protein capsids, and those without envelopes. Okay, so envelopes comes in different forms. Even some of the envelopes will have spike proteins. Okay, but the main important thing about the viral genome is whether their chromosome is linear or chromosomal, and they are not actually fixed. A virus can have a linear or circular chromosome. The genetic material can be DNA or RNA, and the genetic material can also be single-stranded or double-stranded. Okay, so nothing is really certain in viruses. And let's look at bacteria. Okay, let's look at how the different pathway viruses actually go through, like based on conditions when they infect an organism. So in this case, we are going to use a phage, bacteriophage. That's a virus that affects or that infects a bacteria. Okay, and the host in this case is E. coli, and this is the virus, lambda phage. So when we know that when a virus infects a host cell, it injects its genetic material. Okay, so upon injection, based on conditions, right, the genetic material actually chooses between two pathways, the lytic cycle or the lysogenic pathway. Okay, so in the lytic cycle, that's like more straightforward. So um the DNA, the genetic material of the virus actually replicates in the virus and assembles. So it actually takes over the replication machinery of the cell. Okay, stealing proteins and other stuff that it will need for replication to take place. Then it replicates itself, assemble new protein particles. Then you realize it will form new plenty virus in the host. Then it will cause cell lysis. Then the cell lysis will release them to infect other neighboring cells. Okay. The other one is when conditions are not favorable. So in these cases, the genetic material is going to incorporate itself into the host chromosome. Okay, so it forms a prophage, and the prophage is going to lie dormant in the host chromosome. So whenever the host replicates, the distance is like now inherently part of the host chromosome now. So whenever the host chromosome replicates, the virus is part of the daughter cell's chromosomes. Okay, so we can induce lysis in the lysogeny. So we can induce lysis such that the virus or the genetic material of the virus will leave the lytic stage and take form of the lytic stage. Okay, so it will leave the lysogenic state and go through the lytic state. So that is called prophage induction. So let's look at bacterial chromosomes. They're actually more specified. Most bacteria have circular chromosomes and not linear, and in bacteria they just have a single chromosome. Okay, and on the chromosome they have only one. Okay, the diagram is not here. They have only one origin of replication. So in eukaryotes, you come to know we have multiple, but in bacteria they have only one origin of replication. And also in the bacterial cell where the chromosome is found is called a nucleoid. Okay, I'm sure I've mentioned this because they do not have a membrane bound nucleus. Okay, so it is just in the cytoplasm, but the region where they are found is called the nucleoid. So a bacteria can have 1 to 4 identical copies of that particular chromosome. Okay, so most bacteria have just one chromosome. So if you happen to see multiple chromosomes, it's just like a copy of the same chromosome. Okay, it's not like they have multiple chromosomes. So in bacterial chromosome, there are two main types of genes. We have structural genes and intergenic regions. Okay, so the structural genes actually forms the dominant portion of their genome, and those are the encoding genes, those that code for proteins. And the intergenic ones are the non-transcribed DNA regions between the structural genes. Okay, that's how we call them intergenic. So they are between genes. Okay, so I think I've mentioned almost everything about bacteria. So there are also repetitive sequences within the chromosome, but it's not really important. So this is how a typical bacterial chromosome looks like with a single origin of replication. Okay. So now let's look at how the bacterial chromosome is packed to be able to fit into the nucleoid or even the bacterial cell, right? So prokaryotes actually undergo 2 levels of folding. So the first one is the circular chromosome being folded into loops. Okay, so the first one is formation of loops. So and this is actually enhanced by proteins. The proteins were named, but I'm guessing they will be histone proteins. Okay, so this first level is reducing the length or folding the chromosome about 10 times. Okay, so the DNA must be compacted about 1,000-fold. That's for the bacterial chromosome to be able to fit into the cell, it has to be compacted about 1,000-fold. Okay, so each loop is like folding the bacterial chromosome 10-fold, and the number of loops actually depends on the bacteria, okay, or the size of the bacterial chromosome. And the second one is supercoiling of the loops that we formed. Okay, supercoiling can be negative or positive supercoiling. So the individual loops are going to be supercoiled. Okay, and this will further reduce their length but increase their thickness. Okay, so these are the two levels of folding, okay, in bacteria. So let's look at the enzymes that actually play a role in the supercoiling. We have topoisomerase 1 and 2, and of course whenever you hear supercoiling you should know topoisomerases are involved. The topoisomerase 2 is also called DNA gyrase. Okay, and the DNA gyrase is in charge of producing the negative supercoiling. Negative supercoiling is actually so let's say if the DNA strand is twisted in the right direction and you are doing supercoiling in the left direction or the opposite direction, you are doing negative supercoiling. So in supercoiling there will be a point where you twist till all the tension has been released, or you cause unwinding. So if you continue twisting, that's the negative supercoiling. So it's like the thing has been done to the right and you are going left, so there will be a point to come to zero. So it's like someone has counted to 5 already and you want to do negative supercoiling, so you are going to count into the negatives. There will be a point you get to zero. Okay, so negative supercoiling is said to promote unwinding. So at the point where we get to zero, when you are counting backwards, that's when you've caused total unwinding of the DNA strand. But if you are doing negative supercoiling, you have to proceed to the negative region. But the positive supercoiling is like you've already counted to 5 and you want to count to like 15. So you are continuing in the same direction. That's positive supercoiling. So negative supercoiling is done by topoisomerase 2, and it also relaxes positive supercoiling, as I said, because it takes it from the positive side to the negative side. And topoisomerase 1 does the opposite, relaxing negative supercoiling and leading into the positive one. So the action of these two enzymes will govern the overall supercoiling of bacterial DNA. And let's see how the topoisomerase 2 does the coiling. Okay, so this is like the structure of topoisomerase 2, the gyrase. It has two jaws, the lower jaw and the upper jaw. Okay, so the DNA will bind to the lower jaw and it will loop around the lower jaw. So after looping, or after, I don't know if you can call it wrapping, after wrapping around the lower jaw, the upper jaw will clamp onto it. Okay, sorry, the upper jaw will clamp onto it and the strand in the lower jaw is going to be cut. Okay, so if the strand in the lower jaw is cut, the strand up in the upper jaw will be released so that it can pass through and fall within. So it's like you are tying a knot. Okay, but for us to be able to pass it through, we have to cut this one to make way for this one to fall down. After this one falling down, or after the upper jaw releases the strand in it, it will fall into the lower jaw. Then we seal the one we cut. Then we will form one supercoiling. Okay, so that's how supercoiling is done. So we just repeat these ones. So it's like this process we've gone through caused this two negative supercoilings. Okay, so as we move on, like going, we realize the whole chromosome will be supercoiled. So this is just a diagram showing like bacterial chromosomes are normally supercoiled and blah 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 blah. And this is a diagram showing positive and negative supercoiling. And as I said, negative supercoiling promotes DNA strand separation. So now let's move on to eukaryotic chromosomes. They are of more importance. Unlike prokaryotes, our chromosomes are found in the nucleus. Okay, prokaryotes have theirs in the nucleoid. So as I said, our nucleus are very very little. It's like is it micrometers or nanometers? Micrometers in length. Okay, and our chromosomes, if stretched, the entire set of chromosomes will be like 2 meters long. So you have to be tightly packed, right, to be able to fit into the nucleus. So see the chromosome packing. So we packing the chromosome into a nucleus. See it as having a paper, okay, and you trying to fit that flat piece of a paper into like a bottle. Okay, so the circumference of that bottleneck is the nucleus in this case. The paper is too big, but it's flat. Okay, paper is like long, but it's flat. It's not thick. So when we muamua the paper, when you crumble the paper, right, you realize it won't be as big it was, even though it's thicker now. It wasn't. It won't be big like the flat paper. That's what we are going to do there to the chromosome. So realize once we are folding, the number or the size, we'll be using some units. So it's like two folding is like 2 nanometers to 10 nanometers to 300 nanometers to 30 to 300 to 700. One will be like, but as we are folding, we are increasing the size. No, we are not increasing the size of the DNA. We are actually increasing the thickness by reducing the length. Okay, so realize the paper, the normal paper is longer than the crumpled paper, but the crumpled paper is thicker than the normal paper because the normal paper is flat. So no matter what we do to it, it can't fit through in the bottle. It can pass through the neck of the bottle, right? But when you crumble it into a smaller ball, now it is thicker but shorter than the flat paper. So that's what we are going to do to the DNA. So you realize the DNA, as it is being compacted, the unit you are giving to is becoming bigger. So that's not the length of the DNA, but rather the thickness. Okay. So I just said that before we actually talked about the compaction thing. So before the chromosome, what am I saying? Chromosome can be, fitted into the nucleus. It has to be. So the compaction is actually done by complexing DNA with proteins. Okay, and that's how we fold the DNA. And these proteins are called histones. So the DNA protein complex is called chromatin. So we wrapping our DNA around certain proteins to make them more compact is called forms a complex, and that complex is called a chromatin. So okay, so I'm sure we know this. Our eukaryotic genes are very big. Blah 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 blah. So I think this is just expanding on what we said about the C-value paradox, that your genome size won't be this. It's not like a direct representation of how complex your genome is. Okay, so example is in salamanders. So they are, even though they have the same set of genes, one particular species, which one? The Recmondi, the one in B, has like almost 2 times. Its genome is like 2 times bigger than the second one, but they have the same genes. So how come? What's the difference? Okay, so the difference is, one organism might have like repetitive DNA sequences. So these are like the same genes but copies of them. So that does not mean you have extra set of genes, but they say we have the same genes, just that you have many copies of them. Okay, and these repetitive sequences do not code for proteins. It's only the original ones that will code for proteins. So you having more genes does not necessarily mean you are going to code for more proteins than I do, right? So mostly we can attribute this thing to the presence of repetitive DNA sequences. So let's look at how the eukaryotic chromosomes are packed. Okay, let's first look at the organization. So as I said, our chromosomes are linear. Okay, and we have a centromere that's like in the middle of our chromosome, and that separates the chromosome into 2 arms. Okay, and we will get the name of the arms and stuff. But unlike bacterial chromosomes, we have multiple origins of replication. Okay, origins of replication. Yeah. So you realize on the bacterial chromosome it was just a circular something with one origin of replication. So this origin of replication actually plays a role in the bidirectional replication of eukaryotic synthesis, DNA synthesis. Okay, so DNA synthesis can move in this way, another can move in that way. Okay, so the genes of an eukaryotic chromosome are actually found between the we have the centromeres here. We also have telomeres at each end, and the genes, as I said, are found between the centromere and telomeres. So essentially it simply means the genes are found on the arms of the chromosome, but not at the centromere nor at the telomeres. Okay, so what am I not? Centromere sequences are repeating sequences and non-protein coding. So there are no genes or protein coding genes at the centromeres, nor even at the telomeres. Okay. So what the role the centromere plays is it helps bind to the centromere proteins. That's the kinetical proteins. Okay, during spindle fiber formation. So in mitosis, when the spindle fiber forms for the chromosomes to separate, that's another role the centromere plays. Okay. So these are some key features of the eukaryotic chromosome. So in lower eukaryotes, eukaryotes, there are different levels of eukaryotes. In lower eukaryotes, obviously, you have smaller, relatively small genes, and in higher eukaryotes, we have many longer genes, and just a direct implication. That means we have many introns than them. So let's look at the compaction of eukaryotic chromosome. Okay, so I think I've given the introduction already. So let's look at the first level of compaction. So like in the normal form, the normal chromosome, that's not compacted. The thickness of that DNA or the chromosome is 2 nanometers. Okay, so the first level of compaction is folding it into a 10 nanometer fiber. So in some cases you see 10, in some cases you see 11. It's the same level of compaction, or it's the same thing. Okay, so the first one is compacting your DNA or your chromosome into a 10 nanometer fiber. So we are simply wrapping our DNA around a protein complex. Okay, and the repeating structure unit within the eukaryotic chromatin is called a nucleosome. So I'll talk about the nucleosome later. So when we wrap our chromosome around a protein complex, right, we get nucleosomes, and these are like like the subunits, okay, of a chromatin. So the chromatin, let me use this. So you realize this is like a chromatin, and this is a nucleosome. Okay, so that's what a nucleosome is. And this protein complex is an octamer of histone proteins. So octamer means is 8 proteins, but most importantly, the histone proteins are 4. Okay, but each one of the histone proteins they come in 2 copies. That's why we call the protein complex an octamer. So the proteins are H2A, H2B, H3, and H4. Okay, so there are 2 copies of H2A, 2 copies of H2B, 2 copies of H3, and 2 copies of H4. That forms the core, or the octamer protein, where we are going to wrap our DNA around. Okay, there is another histone protein, but that does not form part of the core, and that's it actually plays a role in linking nucleosomes. Okay, and that's the H1 histone 1. So yes, the overall structure of the nucleosome resembles beads on a string structure. Okay, so this is a nucleosome. It's like beads on a string. The string is your chromosome, and the bead is like the protein complex, and this is another nucleosome. So realize this is the thickness of the nucleosome, or of the chromosome at this level. It is 11 nanometers. This is not the length, but rather the thickness. Okay. Histone proteins are actually basic. So we are just trying to establish the association between how the DNA is able to associate with the histone proteins. So the histone proteins are basic, mainly consisting of lysine and arginine tails, and we know basic we have positive charges. Okay, basic amino acids have positive charges, and the phosphate backbone of DNA they bear negative charges. So electrostatic charges are actually unlike negative charge attract positive, or they combine to positive. So the positive charge amino acids in the histones can easily pair or bind with, or let's say interact with the negative charges on the DNA backbone. Okay, so that's one way of improving interaction between the chromosome or the DNA and the proteins into forming the nucleosome. Okay, and as I said, besides the 4 histone proteins that form the octamer, there's another one called the H1, and the H1 plays a role as a linker histone. So this would be like the linker histone. Is it this? Yes, this one, trying to connect different nucleosomes. Okay. So let's look at the second level of folding. Sorry, the second level is further. So I think the first one, I'm coming. Okay, so the first one, by the end of the first level of folding, which is forming the 10 nanometer something, we have by then we would have folded the DNA by 7-fold. Okay, so like we folded like 7 times of its original size. And the second one, which will be like further folding from the 10 nanometer or the 11 nanometer into 30 nanometer fiber, will also further fold it another 7-fold. Okay, so by the end of the second round, you realize you would have folded the DNA almost 50 times. 7 times 7 is 49, so that's like almost 50 times. So the second one is association with each the nucleosomes, the 10 nanometer fibers that we've produced, association of them to form more compact structures. Okay, and this is where the histone 1 plays an important role. Okay, linking the numerous nucleosomes. Okay, so let's look at a particular structure. Okay, it's not on this page. Okay, it's on this page. So actually the structure or the shape of these 30 nanometer fibers are not really certain. So there are two propositions. We have the solenoid model and the 3D zigzag model. Okay, but the 3D zigzag model is like widely accepted as true, unlike the solenoid model. But in the solenoid model, the nucleosomes have more interactions. Okay, so they actually interact more, so they come together to form like a globular protein sort of thing. Okay, but in the zigzag, even though they are brought together, interactions are less. Okay, so this is an irregular configuration where nucleosomes have little face-to-face contact. Okay, but they are brought together by the help of the histone 1, the H1. Okay, the H1 protein. So this is your 30 nanometer fiber. So this nucleosome, this is a nucleosome, this is a nucleosome, this is a nucleosome. Each one of them is like each one of them individually is the first step. That's the 10 nanometer fiber. But when they are brought together, they are compacted further into a 30 nanometer fiber. And mostly you have like 8, I don't know where it says in the slide, but I've seen it somewhere before. 6 to 8 nucleosomes per 10. Okay, so per structure, or every 30 nanometer fiber per structure, or per 10, you get about 6 to 8 nucleosomes. Okay, so that's the second level of folding. So as I said, by then, after the second, you would have folded the DNA about 50 times. So the third one, which is moving on to 300 nanometer, is the formation of loops. So in this case, we call them formation of radial loops. Okay, so in this case, we need interaction between the 30 nanometer fiber that we just formed. That's the 3D something that we just formed in the previous page. We need interaction between them and the nuclear matrix. Okay, so this is like within the nucleus. Okay, actually the nuclear matrix is composed of two parts, the lamina and the proteins. The lamina actually is at the peripheral. Okay, so these brown things are the peripheral, just beneath the nuclear membrane, are called the nuclear lamina, and the yellow stuff are the nuclear matrix proteins. Okay, so we need interactions with these two. Okay, so depending on what the chromosome or chromatin or whatever, depending on what it binds to or interacts with, will determine the nature of the chromosome. Okay, so mostly we come across. Okay, so let me also say this in the next page. The third mechanism, as I said, formation of radial loops. So the chromosomes are in the nucleus are not like just random. They are not in the nucleus randomly. They are found in discrete and non-overlapping chromosomal territories. Okay, and we have two chromosomal territories, the LADs and the TADs, the lamina associated domains and the topologically associating domains. So as I said, between the nuclear lamina and the nuclear matrix proteins, whichever one you find yourself interacting with will determine the type of chromosome you'll be, or chromatin you'll be. So those that associate with the lamina are mostly heterochromatins. Okay, so we come to the difference between euchromatins and heterochromatins, but now you have to put in your head that those that associate with the laminas, the nuclear lamina, form, or they are more compact, okay, and less active, hence they are heterochromatins. And those, I wouldn't say those let me just see, because I've not found it anywhere, but because there are two interactions between the lamina and the nuclear proteins, and if those interacting with the lamina form the heterochromatin, then obviously those interacting with the nuclear matrix proteins are going to be more achromatic than the those with the lamina. Okay, so those that interact with the nuclear lamina are actually found in this territory, the large territory. So the chromosomes within the large territories are mostly heterochromatic, and the chromatins of, am I saying proteins? Sorry. The chromosomes found in the third region, the topologically associated domains, are mostly active. That's like eukaryotic. Hey, and then man, from too much. Euchromatic. I have to go and eat, Charlie. So let's, okay, I'm not sure there's actually a diagram for it, but let's recount. I think this is the last level of folding we are going to discuss, but this is not the last one. Okay, so most euchromat what's the name, euchromatin, yes, most euchromatins actually are in this the 300 nanometer stage. Okay, they don't go further, but for further compaction, we will fold the 300 nanometer into a 700 nanometer chromatin, right? Then two sister chromatids will come together to form your chromosome, and that chromosome is said to have a thickness of 1400, because two 700s come together to form 1400. Okay, so let's use the diagrams on these two pages, this page and the next, to just summarize everything we've said about chromosomal compaction or packing. So this would be a normal DNA. You see, it is very long, but it is not thick. The thickness is 2 nanometers, right? And the first level of compaction is wrapping it around histone proteins. That reduces the length and increases the thickness to 10 nanometers. The second level is coming together of these nucleosomes, so to form the 3D zigzag structure, and this increases the diameter or the thickness to about 30 nanometers, right? But in this case, what was I even going to say? Charlie, my mind is not here today. I forgot what I was about to say. But you should know that in this case we would have folded the proteins about 50 times, or let's say 49 fold, right? Then after the 30 nanometers, we form radial loops into the 300 nanometer structure. So these ones, you see there are no proteins here. That doesn't mean there are no proteins. Actually, the protein should be here, but probably because the diagram are not showing them doesn't mean there are no proteins. The proteins are still there, histones are still here, but we form loops, right? So you form loops. So the as I said, euchromatins mostly are in these forms. Okay, so and these actually happen during the interface region, like during cell division, mitosis, and those stuff. Okay, and if you are going to further compact it, we are going to do like further compaction of the radial loops, so that's more of like supercoiling stuff. Okay, so these are tightly packed, unlike the heterochromatin. That makes them less active. Okay, so that would be the 700 nanometer, and that would be like one sister chromatid. Then it can line with another sister chromatid to form the 1400 nanometer chromosome. Okay, and this is called a metaphase chromosome. So let's look at the differences between heterochromatin and euchromatin. As I think I've already mentioned enough about them. Euchromatins are less condensed. Okay, so from this diagram, the diagram before us, you can tell. Okay, so the euchromatin actually mostly end at the 300 nanometer stage. Okay, so they are less compact and they are transcriptionally active. So that means they can be transcribed. Okay, and the regions where, so those are where we form the radial loops, and in the heterochromatin they are actually tightly compacted. That means the radial loops are further compacted. Okay, and they are transcriptionally inactive in general. There are two forms of the heterochromatin. We have constitutive and facultative. Constitutive is heterochromatic regions where that like it's like they are permanent. They are always heterochromatic. Okay, that means they are permanently inactive. But for the facultative, they can change between heterochromatin and heterochromatin. Okay, so on a normal chromosome, let's say on a euchromatin, the arms of the chromosomes can be euchromatic, but the telomeres and the centromere can never be euchromatic. Okay, so the centromere, telomeres, and the telomeres are like the constitutive heterochromatins for them, because we don't need them to code for anything, so they are permanently inactive. Okay, so let's look at some proteins, major proteins that are involved in the chromosome structure. Okay, I'm sure you know what the histones does. The linker protein, which is another histone, and a protein called scaffold. The scaffold forms part of the skeleton of the condensed chromosome. Okay, and the kinetochore, I did mention this. It forms part of, it is a centromere protein. Okay, it is found at the centromere, and it helps in cytoskeleton attachment to the centromere. Okay, mostly during spindle fiber formation. And I'm sure we know the telomerase. We also know telomere caps. Okay, so this will be the end of part one. In the next part, we are going to look at different methods in the distinguishing between euchromatic regions and heterochromatic regions of a genome. Okay, so I'll see you you guys in the next one.